All right, let's talk about one of the oldest and honestly most powerful parts of your immune system. You've got this silent guardian, this first responder that's always on patrol in your bloodstream. And today, we're putting it right under the microscope.
So have you ever wondered how your body handles an infection before your main immune cells even get the memo? Before your T cells and B cells even know there's a problem. How do you fight back in those first critical moments? Because there's no time to wait for backup. That first wave of defense, it has to be immediate and decisive.
Well, the answer is it calls in a fast-acting security detail, the complement system. I want you to think of it as both the security alarm and the sprinkler system for your body, all rolled into one. This is a key player in our innate immunity, the defenses we're all born with.
Okay, so we're calling it immunity's secret weapon. But what is it exactly? Let's dive in and take a look at the arsenal it brings to the fight. So, the complement system. The name complement is actually perfect because that's exactly what it does. It's this whole group of more than 30 proteins just floating around in your blood, basically waiting for a trigger. They don't replace your other immune cells. They complement them. They assist them, making their jobs way faster and a lot more effective. And this system is, wow, it's incredibly versatile. It's got a few core functions. First off, it can directly attack and just burst invaders. That process is called lysis. Second, it acts like a flare gun, promoting inflammation to call other immune cells to the scene of the crime. And third, it can tag pathogens for destruction, a process called opsonization, which basically makes them an easy meal for our cleanup cells. It really is the perfect bridge between our immediate innate defenses and our long-term adaptive ones.
All right, a system this powerful has to have a reliable way to get switched on, right? But it's not just one single alarm button. Turns out there are actually 3 distinct pathways to kickstart this whole process.
Okay, I know this diagram might look a little complicated at first glance, but the key takeaway here is just how efficient it is. See how all 3 of those different pathways, the classical, lectin, and alternative, they all funnel down to one crucial component, a protein called C3. It doesn't matter how the alarm gets triggered, the final response, the inflammation, the tagging, the destruction, it all stems from this one single hub. So what this really boils down to is the trigger. The lectin and alternative pathways, they're part of that immediate, innate response we talked about. They can recognize patterns directly on microbial surfaces. But the classical pathway, that one's a little different. It gets activated by antibodies that have already flagged an invader. And that's huge, because it means this pathway links up with our adaptive immune system, the one that learns and remembers specific bad guys. All right, now let's zoom in and see how this whole thing actually works. We're going to focus on the classical pathway as our main example, so we can really see this amazing chain reaction, this complement cascade, in action. Yeah, thinking of it as a domino cascade is the perfect analogy. See, these proteins are just floating around as what are called zymogens, which is just a fancy word for inactive precursors. They're like dominoes, all set up and just waiting to be tipped over. And once that first one falls, it sets off this incredible chain reaction where each step activates and then massively amplifies the next one. So we can break this whole cascade down into 4 key stages. We've got the trigger, then we build the engine, then we hit that crucial hub for amplification, and finally we create the enzyme that's going to launch the final attack. Okay, step 1, the trigger. Here's what happens. An antibody, let's say an IgG, has found an invader and latched right onto it. The very first complement protein, which is called the C1 complex, recognizes this antibody-invader pair and binds to it. That's it. That's the official start signal. The first domino has just been pushed. Step 2 is about building the engine. As soon as C1 is activated, it basically becomes an enzyme. Think of it like a tiny pair of molecular scissors. Its job is to find and cut 2 other proteins, C4 and C2. Then, specific pieces from that cut, C4b and C2a, they join together to form a brand new, really powerful enzyme. This is the engine of the cascade, and it's called C3 convertase. And now we get to that crucial hub, step 3. That C3 convertase we just built, it goes to work on the most abundant complement protein in your blood, which is C3. And I mean it goes to work. It's a cleaving machine, just splitting thousands upon thousands of C3 molecules into 2 parts. You get C3a, which floats away to act as that inflammatory flare, and C3b, which sticks right to the pathogen, tagging it for destruction. This is that massive amplification step we were talking about. And here's where it gets really clever. For step 4, we're creating the final enzyme. The process just builds on itself. One of those C3b fragments that was just created actually joins up with the C3 convertase that made it. And this one simple addition completely changes the enzyme's function. It turns it into a new enzyme, C5 convertase. And its only job is to kick off the final destructive phase.
And that final phase? Well, it has a truly spectacular and honestly perfectly fitting name. This is the ultimate goal of the entire cascade, the membrane attack complex.
The goal here is beautifully simple and kind of brutal. The whole endgame of the complement system isn't to poison or disable the pathogen. Nope. It's to punch a hole right through its cell membrane.
So here's how it happens. That C5 convertase we just made starts cutting up C5. The C5b fragment that's created then drills itself into the enemy cell membrane, acting like an anchor. And from there, it starts recruiting a whole sequence of other proteins, C6, C7, C8, and finally a whole ring of C9 proteins. They all come together to assemble this incredible structure you see here, the membrane attack complex, or MAC for short.
What this MAC really is a hollow cylinder. It forms an open pore right through the pathogen's membrane. And with that hole punched in it, water and salts just rush in uncontrollably. The cell swells up like a balloon and, pop, it bursts. That's called osmotic lysis, a very direct and very, very final end.
Okay, so with all of this destructive power, you have to be wondering, what happens when the system fails? What could possibly go wrong? Well, the answer is quite a lot, and in a couple of very different ways. Which leads to the most critical question, right? With such destructive power, what stops the complement system from attacking our own cells? If this whole cascade can get started on a microbial surface, why doesn't it just run wild and start punching holes in our own healthy tissues?
Well, thankfully, our bodies have some really clever built-in safety controls. We've got soluble regulators like C1 inhibitor just circulating in the blood, ready to shut down the cascade right at the beginning. And even more importantly, our own cells have special proteins on their surfaces, like DAF and CD59, that basically act as don't shoot me signals. They make sure activation is localized and short-lived by actively dismantling any complement components that happen to land on them. And you can see the direct and sometimes devastating consequences when just one piece of this system goes missing. For example, a deficiency in an early component like C1, C2, or C4 can lead to autoimmune issues like lupus. If you're missing C3, that central hub for all pathways, that's catastrophic. It leads to severe or current infections. Or if you're missing a piece of the final MAC, you become highly susceptible to specific bacteria like Neisseria. It's just an incredibly clear link between a single protein and a specific disease. So we're left with this really interesting idea. This ancient defense system is a total double-edged sword. It's elegant, it's incredibly powerful, but when it's out of balance, it can be a major driver of disease. So the real challenge for modern medicine now isn't just understanding it, it's learning how to control it. How do we fine-tune it? How can we turn it up to help fight things like cancer, or turn it down to treat autoimmune disorders? It's a truly fascinating puzzle left for us to solve.